Hemos becado a más de 1 millón de estudiantes universitarios.
Más de 84,000 alumnos y alumnas han sido beneficiados con becas nutricionales.
Lanzamos una campaña para apoyar a la Universidad Autónoma de Guerrero, afectada por el huracán Otis.
Este año organizamos nuestra tercera gala musical y nuestra cuarta carrera atlética.
Estamos felices porque más del 87% de nuestros becarios son la primera generación de universitarios en su familia.
Cada año se apoyan a 100 estudiantes de la Facultad de Derecho.
Hemos entregado más de 600 becas a estudiantes de bachillerato junto con Fundación BBVA.
Hemos transmitido 195 documentales y 160 conferencias.
Apoyamos a la Unidad de Investigación en Neurodesarrollo de la UNAM.
Todos nuestros informes son públicos.
Se han entregado más de 50 premios para fomentar la investigación científica.
Con el programa Bécalos, el Patronato de la Facultad de Química y Fundación UNAM otorgaron más de 30 becas.
Con la ayuda de Fundación Carlos Slim hemos otorgado becas de apoyo nutricional y económicas.
Con el programa Hídrico Modelo ayudamos a 340,000 habitantes.
Tenemos 2 premios EFI Social por nuestras campañas publicitarias.
Junto a Grupo VAL, más de 2,000 universitarias han sido beneficiarias.
Otorgamos más de 400,000 tratamientos.
Durante la pandemia apoyamos con las campañas Dona un Kit y Apoya a un Residente a estudiantes de medicina de la UNAM.
Durante el confinamiento COVID, donamos tablets para estudiantes de escasos recursos.
Hoy son más de 27,000 solidarios impulsando la educación.
Participamos en la conservación de corales en el Caribe mexicano.
Brindamos más de 4,000 becas de movilidad internacional.
Colaboramos con el Semillatón a preservar el maíz en la Sierra Tarahumara.
Apoyamos el proyecto de conservación de perritos llaneros en la Reserva de la Biosfera de Jano, Chihuahua.
Participamos en el proyecto de conservación de la guacamaya roja en el Parque Nacional de Palenque.
Desarrollamos el programa Líderes Transformadores.
Participamos con el Consorcio de Universidades por la Ciencia.
Organizamos 11 ediciones del Foro 2020.
Fundación UNAM resguarda el Palacio de la Autonomía, inmueble histórico de la universidad.
A través del CI ofrecemos cursos, diplomados y talleres.
Dotamos de equipo al Observatorio Astronómico Nacional de San Pedro Martes.
Apoyamos a alumnos de escasos recursos.
Asignado en la opción señalada, institución de asignación UNAM.
¡Sí quedé!
El sueño se cumplió. Ahora formas parte de la comunidad universitaria y puedes acceder a las becas que Fundación UNAM tiene para ti. Conoce nuestros programas en www.funam.mx y en nuestras redes sociales.
Hemos becado a más de 1 millón de estudiantes universitarios.
Más de 84,000 alumnos y alumnas han sido beneficiados con becas nutricionales.
Lanzamos una campaña para apoyar a la Universidad Autónoma de Guerrero, afectada por el huracán Otis.
Este año organizamos nuestra tercera gala musical y nuestra cuarta carrera atlética.
Estamos felices porque más del 87% de nuestros becarios son la primera generación de universitarios en su familia.
Cada año se apoyan a 100 estudiantes de la Facultad de Derecho.
Hemos entregado más de 600 becas a estudiantes de bachillerato junto con Fundación BBVA.
Hemos transmitido 195 documentales y 160 conferencias.
Apoyamos a la Unidad de Investigación en Neurodesarrollo de la UNAM.
Todos nuestros informes son públicos.
Se han entregado más de 50 premios para fomentar la investigación científica.
Con el programa Bécalos, el Patronato de la Facultad de Química y Fundación UNAM otorgaron más de 30 becas.
Con la ayuda de Fundación Carlos Slim hemos otorgado becas de apoyo nutricional y económicas.
Con el programa Hídrico Modelo ayudamos a 340,000 habitantes.
Tenemos 2 premios EFI Social por nuestras campañas publicitarias.
Junto a Grupo VAL, más de 2,000 universitarias han sido beneficiarias.
Otorgamos más de 400,000 tratamientos.
Durante la pandemia apoyamos con las campañas Dona un Kit y Apoya a un Residente a estudiantes de medicina de la UNAM.
Durante el confinamiento COVID, donamos tablets para estudiantes de escasos recursos.
Hoy son más de 27,000 solidarios impulsando la educación.
Participamos en la conservación de corales en el Caribe mexicano.
Brindamos más de 4,000 becas de movilidad internacional.
Colaboramos con el Semillatón a preservar el maíz en la Sierra Tarahumara.
Apoyamos el proyecto de conservación de perritos llaneros en la Reserva de la Biosfera de Jano, Chihuahua. Participamos en el proyecto de conservación de la guacamaya roja en el Parque Nacional de Palenque.
Desarrollamos el programa Líderes Transformadores.
Participamos con el Consorcio de Universidades por la Ciencia.
Organizamos 11 ediciones del Foro 2020.
Fundación UNAM resguarda el Palacio de la Autonomía, inmueble histórico de la universidad.
A través del CI ofrecemos cursos, diplomados y talleres.
Dotamos de equipo al Observatorio Astronómico Nacional de San Pedro Martes.
Apoyamos a alumnos de escasos recursos.
Bienvenidos al ciclo de conferencias de investigación Universidades por la Ciencia, organizado por Fundación UNAM, el Colegio Nacional y el Consorcio de Universidades por la Ciencia. Les agradecemos el interés y que nos acompañen. Y el día de hoy tenemos una conferencia a cargo de la doctora Maanasa Raghavan de la Universidad de Chicago, dentro del ciclo de los estudios de genómica y paleogenómica y la evolución humana. Este ciclo ha resultado muy interesante y, como hemos recalcado desde el inicio, esta área de investigación se caracteriza por un muy rápido desarrollo. En tan solo unos años, la resolución de estos estudios para investigar la firma genética, nuestro código de vida, ha revolucionado. Es realmente impresionante la forma en que el campo se continúa desarrollando y las perspectivas son todavía mucho más interesantes. El ciclo es organizado por las doctoras Mashaal Sohail y Araxi Urrutia del Centro de Ciencias Genómicas y del Instituto de Ecología de la UNAM. Y en el panel nos acompaña el presidente de Fundación UNAM, Dionisio Meade, nuestra presidenta del Consorcio Universidades por la Ciencia, Araxi Urrutia, y nuestra conferencista invitada, Maanasa Raghavan. Welcome to the seminar cycle on genomics and paleogenomics, organized by Mashaal Sohail and Araxi Urrutia. This conference, this seminar cycle has been organized by the UNAM Foundation, the Colegio Nacional, and the Consortium of Universities for Science, with universities for many different places in the USA, Canada, UK, Spain, Brazil, and in the USA, we have also the strong participation of the University of Chicago. Maanasa is a professor in the Department of Human Genetics in the University of Chicago. So we are very glad having this conference, and the presentation will be made by Dr. Mashaal Sohail, and we have just an introduction by our president of the UNAM Foundation, Dionisio Meade. welcome everyone, and we invite you to send comments, questions to the platforms of the Universities for Science Consortium. Welcome everyone. Dionisio, por favor, and
muchas gracias, Jaime. Muchas gracias a todos por acompañarnos en esta nueva conferencia vinculada a la genómica, paleogenómica, que tiene que ver pues con la evolución y una explicación de lo que ha pasado con todo eso. Para nosotros es muy grato que se celebre este evento y, como lo hemos mencionado en otras ocasiones, básicamente nuestro propósito es que nuestra audiencia, mucha de ella integrada por jóvenes que están por definir su perfil profesional, encuentren en la investigación científica, en estos campos puntuales que están siendo objeto de tratamiento en estas pláticas, un campo de realización profesional que justamente les permita realizar esta opción y puedan encontrar que la vida de la ciencia es una vida apasionante, es una vida que ofrece enormes atractivos y es una vida que ofrece también la realización de una vocación de investigación y profesional. Muchas gracias de nuevo. Agradecemos en esta ocasión a Maanasa, que nos hace el favor de acompañarnos desde la Universidad de Chicago. A nosotros nos da mucho gusto porque en Chicago también tenemos una oficina de la UNAM y también pretendemos que se sume la audiencia de cada uno de estos centros en donde nosotros tenemos presencia a escuchar las pláticas y las presentaciones que se hacen. Mashaal ha venido organizando junto con Araxi una serie extraordinaria y queremos agradecer de manera particular a nuestras propias autoridades universitarias y a las del Colegio Nacional, porque juntos hemos venido realizando este esfuerzo de divulgación y de información para que podamos reconocer su esfuerzo, su participación y agradecerles también que estén con nosotros. Quiero mencionar que en esta década, primero fue un año, ahora es una década de investigaciones, de promoción de la UNESCO y de Naciones Unidas para impulsar la investigación de las ciencias básicas. Se nos ha invitado a nosotros como integrantes del comité asesor, justamente por estimar que pláticas como estas son un vehículo adecuado para divulgar la investigación científica. Así que eso nos genera también una gran sensación de orgullo y bienestar, pero también una gran responsabilidad para que podamos mantener a la altura estos esfuerzos de divulgación en los que hoy estamos participando. Muchas gracias a todos. Gracias a Maanasa por esta presentación que nos va a hacer. Gracias a Mashaal y Araxi por organizar este evento y gracias a Jaime por sus palabras de arranque a esta presentación y por su apoyo invaluable para la participación también tanto de la UNAM como del Colegio Nacional para la realización de este evento. Muchas gracias a todos y muy buenas tardes y con la mejor disposición de oír tu presentación. Gracias a todos.
Muchas gracias, Jaime. Dionisio, buenas tardes a todas y todos. Hoy nos vamos a escuchar de la doctora Maanasa Raghavan. Maanasa Raghavan is an assistant professor in the Department of Human Genetics at the University of Chicago. La doctora Maanasa es profesora asistente en el Departamento de Genética Humana de la Universidad de Chicago. She did her bachelor's in molecular biology at the University of Toronto, her master's in archaeological sciences at the University of Oxford, and her PhD in paleogenomics at the University of Copenhagen. Ella hizo su licenciatura en biología molecular en la Universidad de Toronto, luego una maestría en ciencias arqueológicas en la Universidad de Oxford y un doctorado en paleogenómica en la Universidad de Copenhagen. Her group's research interests span questions and applications in multiple fields, including population genomics, anthropology, archaeology, and medical genetics. The big question driving her research is how have demographic, cultural, and environmental factors contributed over time to shaping the genetic profiles of present-day human populations? In order to address these research questions embedded within this goal, her lab uses a combined approach that brings together genome-scale data from present-day and ancient humans and their biotic environment, especially domesticates and pathogens. He's also committed to advancing ethical and sustainable research practices in paleogenomics and to science outreach and communication, featuring both in local initiatives in Chicago as well as documentaries. Today, she will speak to us about genetic insights on human evolutionary histories, case studies from around the world. In this cycle, we started in Africa, where all human history began. Then we learned about present-day human genetic structure, paleogenomics, what paleogenomics has taught us about the history of Europe and of Mexico. Today, we will circle back to see what paleogenomics and present-day DNA can tell us about evolutionary around the world. Pues la doctora Raghavan, sus intereses de investigación abarcan preguntas y aplicaciones en múltiples campos, como la genética de poblaciones, la antropología, la arqueología y la genética médica. La pregunta clave que impulsa su investigación es cómo han contribuido los factores demográficos, culturales y ambientales a lo largo del tiempo a moldear los perfiles genéticos de las poblaciones humanas actuales. Utiliza su laboratorio un enfoque combinado que reúne datos a escala genómica de humanos actuales y antiguos y su entorno biótico, especialmente animales domésticos y patógenos. También está muy comprometida con el fomento de prácticas éticas y sostenibles en paleogenómica, así como la divulgación y comunicación científica. Hoy nos hablará sobre qué pistas nos puede dar la genética sobre la historia evolutiva humana, perspectivas de estudios globales. En este ciclo comenzamos en África, donde empezó toda la historia humana. Luego aprendimos sobre la estructura genética humana, la paleogenómica, lo que la paleogenómica nos ha enseñado sobre la historia de Europa y de México. Y hoy volveremos a analizar qué la paleogenómica y el ADN actual nos enseñan sobre las historias evolutivas en diferentes partes del mundo, usando estudios específicos locales. Muchas gracias, Maanasa. Thank you so much, Maanasa, for being here. It's a great pleasure to have you. So we're very excited about your talk.
Fantastic. Let me just start by sharing my screen. And as I'm doing that, let me take this opportunity to thank all the organizers, especially Mashaal, who reached out to invite, and the amazing introduction that you've all given me. Let me just hide this. Okay, so I will spend the next 30 to 40 minutes almost taking you all on a world tour of sorts. And it's kind of nice to be at the tail end of the series, because I feel like the previous speakers have all set very great context about generally genetic research, be that with modern DNA or with ancient DNA. And what it's sort of added to our understanding of our species, but also perhaps other species. And so I'm going to kind of take a page and expand a little bit in today's talk, and maybe touch upon other regions of the world that maybe haven't been discussed before in this seminar series. I took on a very ambitious title. It says global case study. So, and then when I started putting together the talk, it came to my attention that there's a lot to cover. So obviously I've been quite selective. So I want to sort of present certain case studies and just get the sort of juices flowing, so we can kind of get into a nice discussion about, how our species sort of originated and spread across the globe. So the title that I gave was Genetic Insights on Human Evolutionary Histories, Global Case Studies. In particular, what the mechanism or one of the evolutionary processes that I'm particularly going to focus on today are migrations. Also in the genetic literature, we refer to it as gene flow, essentially people moving and how that sort of shaped the genetic variation and genetic structure that we see today. So just kind of taking the big picture here, when we think about how people migrate and what sort of dictates where people end up, or when people think about moving from one place to the other. Obviously there's many factors in play, but one can think about several of these factors. For example, ecology and geography. So when people move, obviously they tend to move towards better resources, right? And along the way, if they encounter, say, barriers like mountains or big water bodies, that's going to shape the way and the route and the direction and even the pace of a migration. Similarly, you can think about migrations when people move. Obviously, today there's a lot of globalization. People move to find new jobs from city to city or even country to country. Similarly, even in the past, in our species history, humans have moved around, in particular to find better regions where they could either find better cultivatable lands or perhaps in search of prey if they were a foraging and gathering society. So subsistence could also, have been a trigger or has been a trigger for human movements. And on the other end, when you think about sort of who moves or how sort of the migratory landscape is shaped, one can also kind of think about things like cultural factors, like languages and even religion, because, people tend to move as a group, and when they tend to move as a group, they tend to kind of seek out people who have similar cultural facets or aspects to their histories. So people who tend to speak the same languages may congregate by nature's side. People who profess the same religion, for example, could would find each other. So there's many cultural, geological, economic and other kinds of factors that shape how and who decides to move and where they end up. And in general, migrations have been really at the core of, thinking about how our species originated and moved around. And this has been, particularly when we think about how this has shaped our species history in terms of evolution, it has been sort of the core of many disciplines who've come together or in isolation study this for many decades. And these include anthropology, archaeology, history, genetics, and also community knowledge and oral history. So a lot of particularly indigenous communities across the globe have had oral histories passed on over generations that may inform or that may have some semblance of knowledge that connects to certain origin places or perhaps relationships to other communities, and that could inform sort of who they may culturally or even biologically share affinities with. Of course, today's talk will focus on the genetic aspect, but I do want to kind of underscore this, and this will kind of come time and again throughout my talk, that again, genetic studies, particularly when we're looking into human evolution and human histories, must be framed within a multidisciplinary sort of design. Genetics alone can't inform the nuances and the details and the complexity of human histories and human migrations over time. And so particularly today, a lot of these fields tend to come together in interdisciplinary fashion to kind of build narratives that may be closer to the truth. And that only happens because they can leverage rich insights from each other. So humans, we move today, obviously there's heavy globalization, but humans have moved around in sort of, throughout human history, right? Since we've been able to trace back the origins of humans to Africa here, humans originated about 200 to 250,000. And when I say humans, I mean anatomically modern humans. So our sort of species. We originated in Africa about 200 to 250,000 years ago. And since then, there have been sort of a range, what we call range expansions or movements of people, initially out of Africa and eventually sort of moving to different corners of the world at different time periods. A lot of the sort of information about when humans arrived in different parts of the world come mostly from archaeology, where either human remains or cultural remains have informed when particular human groups would have made it to a particular region of the world. And needless to say that, these migrations in the past as well, and now we know better through the studies of both ancient and modern DNA, that, even when you think about these different migration waves that have sort of gone all over the world, a lot of times they're not in isolation themselves. So a lot of time, even these ancient human groups who migrated may have met and mixed with each other. So these movements and the meeting of people and the mixing of genes have really, been around in our species history for a long, long time, as you can see from this map. In particular, the sort of the rich knowledge that we've gained on human movements in the past have come particularly in the last, I would say, about decade or 15 years. And that has sort of been closely tied with this technological revolution called ancient DNA or paleogenetics or paleogenomics. I'm sure a lot of the previous speakers have spoken to you about it, so I'm not going to get into technical details, but I just want to impress upon you just how big an impact this technology of ancient DNA has had in our studies of our own species past. So you can see the sort of exponential rise in the number of human genome data sets that we've had, and this sort of really shoots up in the past 10 years. Of course, a large proportion of these genetic data sets do come from Europe in particular, and we still have underrepresentation, as you can see also in this map, from many other regions of the world, particularly the tropics, where preservation of DNA can be challenging, but also in several regions of the world where colonial histories have played a role, and sort of marginalization of indigenous communities has led to stricter regulations around human, at least ancestral human remains research, for example, in the United States and Canada. Nevertheless, we've still been able to glean a lot from these ancient DNA data sets, even though a majority of them do come from Europe and sort of Eurasia broadly today. But one of the things that have become amply clear is that humans have partook in migrations at different temporal and spatial scales over time, right? So we're looking at ancient migrations, and here I'm just showing a couple of examples, and we're going to go through some of these examples in detail in a bit. But again, from this map, we can see that almost starting about 50 to 60,000 years ago with the out of Africa migration, all the way to that a few thousand years ago, whether that's sort of the different peopling of the continent, so that'll be more long range, or we can think about more locally within, let's say, the European context. This is signifying the steppe-related expansions, and we'll talk about that as well in a bit. That happened about 5,000 years ago, that sort of expanded from the steppe region into Europe. And again, these may not be short-range distances, but obviously I'm contrasting, these are all relative to one another. So from the sort of peopling of the continent scale to more, more of a limited range. Obviously, we can go even further fine scale in terms of the geographic range today, with really fantastic insights that ancient DNA has provided on these different spatiotemporal ranges. On the other end of the temporal scale, then we can also come to very, very recent times, like I mentioned. So here we're looking at the last few hundred to 1,000 years or so. And again, different methods in genetics, in population genetics exist for this, both for dealing with modern DNA data sets as well as ancient DNA data sets. In this particular case, the example I've pulled here is leveraging what are called haplotypes. So these are essentially a string of mutations that sort of occur on a DNA stretch, and you can kind of leverage that to make more powerful inferences in more recent times, and that's what sort of this is doing. So you can really pick out recent timescale related, both short and long range migrations. For example, the migrations related to the colonization of the Americas from Europe or from Africa related to the transatlantic slave trade, as well as more kind of within continental scale migrations, such as the Bantu speaking migrations within Africa. So I just kind of wanted to set the scene here to get you to appreciate how complex and how sort of how different scales of migration experiences can sort of come into play when we think about what shaped the human genetic variation and population structure across global scales. So now I want to kind of just go through several case studies as the title promised. And again, we'll touch upon different temporal scales. We go all the way from out of Africa migration about 60,000 years ago to more recent migrations, and we'll also go from long to more sort of regional migrations. As I mentioned a few slides back, human origins are in Africa. This has been fairly well established, both from paleoanthropology / archaeology, as well as genetic studies. And what has also been sort of brought to light, again, from multiple fields, is that about 60,000 years ago, there was a large migration out of Africa. And so this is sort of what brought many of the ancestors of present-day non-African populations to come and sort of people the rest of the world. And you can kind of see that in this sort of more recent time scales where you have different, in this case, continental scale sort of peopling events that have happened in the last 50,000 years. But I want to kind of focus first on this very, very key migration in our species history, which is this out of Africa migration. And even from modern genomes, in fact, a lot of what we know from the genetic perspective on the out of Africa migration actually comes from modern or contemporary genetic data, because again, as discussed, very few ancient genomes exist from this region of Africa, particularly none that exist that overlap with this time period, and that's particularly because of the extremely tropical, hot and humid conditions across Africa, which means there's going to be very limited DNA survival, particularly as we push back in time. But even when we look at modern human genomes, you do see this very strong signal that this out of Africa migration has left in the genomes of non-African populations. So what I'm showing here is a plot that essentially takes you through a proxy of population size. So in genetics, it's a jargon called effective population size, but for the sake of this talk, you can kind of think about this as if you go up here, that means an increase in population size, and if you come down, that means a decrease in the population size. And on the x-axis here, both on the bottom and the top, we have time. So I think it would be easier for you to look at the top end of this axis because that's the calendar year. So we go from ancient times, about 600,000 years on this end, to more recent times, about 7,000 years ago or so. So we're looking at essentially how population sizes have changed over the course of the last 600,000 years in the history of our species. And we can do that by leveraging genomic data from different modern human populations, as was done in this paper, from a seminal data set that's been sort of used a lot and very pervasive in human genetic studies. It's called the 1,000 Genomes Project, which back in 2015 was their first flagship paper where they published several modern human populations that were sampled from different continents to kind of survey the genetic diversity across the world. So each of these curves represents a modern population, and for the sake of sort of the message I want to give out here, what I want you to focus on is all the yellow curves belong to populations or individuals from populations in Africa, whereas the other colors belong to non-African individuals. And if we come to sort of this seminal out of Africa migration that we've been talking about, that happens somewhere in this time frame. So we're talking about sort of 60 to 100,000 years. And you can sort of see that at that point, you see a massive decline in both African and non-African populations, but the decline of population size is much more significant in the non-African populations around this time. And over time, all populations seem to sort of recover, but this sort of, this sort of massive dip in the population size is taken to indicate the fact that when humans moved out of Africa, so a small set of ancestors moved out, and so that essentially meant that the rest of the world was essentially peopled by a subset of the diversity of the ancestral human gene pool that lived in Africa back in time. Other ways to look at this, again, from the same 1,000 genomes data set, is to look at how much variation can you see in different worldwide populations. And again, when we look at sort of on the y-axis a proxy for how much genetic diversity there is in these populations, you can sort of see that some of the highest diversity in terms of genetics is represented in African populations. Again, the color scheme is the same, so yellow populations or yellow symbols are all African populations. Whereas the rest of the world, you can see a very, very significant difference in terms of the genetic diversity that they carry vis-a-vis the African populations. And again, this sort of comes from the phenomenon that Africa represents the homeland of the original diverse ancestral pool for all the other humans who derive ancestry then from the sort of broader African gene pool. Again, sort of different visualizations, but I think it's really, really cool to go through some of these visualizations to really get at this point. Another way to look at this is when you plot distance from Africa versus genetic diversity here. You can kind of see that you start with African populations here in black, and as you move away from Africa and come all the way to the Americas, you're sort of reducing in that genetic diversity. And again, this is sort of in sort of population genetics jargon, this is something called a founder event or a series of founder events, where again, you can imagine that there's a very diverse gene pool of ancestors in Africa. And as some of them migrate out, they may sort of take a few of these colors which represent genetic diversity with them. So in this case, you can see the yellow sort of gene or the yellow color has been completely lost. And that's because of the randomness of who decides to migrate and have offspring in the next generation. And again, as you take a subset of this diversity who decides to then move on, you lose the red color and so on. So the sort of serial loss of genetic diversity happens when you move further and further away from the source or the origin. And it's a very, very classic kind of signal that we see in human genomes today, which is really a product of these migrations out of Africa and subsequent events. So once humans come out of Africa, there's sort of this other phenomenon, and I don't know how much has been talked about this in the previous previous series. So I thought I would put in a little bit on this because it's quite exciting to think about human admixture histories in this sense, and that is when sort of the subset of humans came out of Africa 60,000 years ago, who did they encounter? And so this has really shaped the way that paleoanthropology and genetics has thought about human origins. And there have been sort of these competing models in particularly paleoanthropology about what our species origins are. And these are sort of almost held two extremes and the sort of the spectrum in between. So you have something called a multi-regional model on one end, which posits that humans originated in Africa about 1 to 2 million years ago. There was this movement, and a likely candidate for this may be Homo erectus back, if you think about the time, 2 million years ago. And they sort of, these early human forms sort of went and peopled the different continents. Eventually, there was sort of gene flow to make sure that there's sort of no, speciation, and sort of populations in these different, continents today are essentially descendants of these different human forms who went back, 1 to 2 million years ago and kind of stayed in those continents. The other extreme of this model of human origins is the recent out of Africa replacement model, and that posits that there may have been obviously several migrations that went out of Africa, which perhaps brought other forms of humans like Neanderthals and Denisovans. But on the other hand, the ancestors of anatomically modern humans, so us, only came out much more recently in contrast to the multi-regional model. And that would have happened, as we just discussed, about 60 to 100,000 years. And then sort of the peopling of the continents happens in the last 50,000 years or so. And so within sort of this framework, there's been a lot of discussion about when humans, move out of sort of Africa, what happens, who do they encounter, and how does that impact, if at all, our genetic history? And so I just wanted to unpack that a little bit, because when humans did move out about 50,000 years ago into Eurasia, there were several other species that were co-inhabiting at that region at that time across Eurasia. But one of the primary sort of species that was hanging around, especially in that sort of region of Europe into East Asia, were the Homo neanderthalensis, or the Neanderthals. And for a long time, there's been this debate about how was there any interaction between anatomically modern humans that just came out of Africa and these Neanderthals, who are, of course, now extinct. And so different hypotheses have been floated, some being that, in fact, modern Europeans are direct descendants of Neanderthals. Other people have postulated that perhaps Neanderthals contributed certain genetic sort of features to modern humans and then died out. Or on the other extreme is that maybe Neanderthals went completely extinct with absolutely no contribution to modern human or our genetic histories. And this has been answered back in 2010 by a team from Germany led by Svante Pääbo, for which he actually received the Nobel Prize in Medicine a few years back. And the answer to this question came from the DNA sequencing of Neanderthals, so the direct sequencing of DNA from these extinct hominins. And the model that sort of came to light from these studies was that yes, indeed, Neanderthals had contributed to modern humans, but in fact, they had only contributed to non-African populations. And so this admixture would have likely happened when humans moved out of Africa. And somewhere in the Middle East, perhaps, we still don't know exactly where or any of the dynamics because we don't have ancient DNA data from that particular time period. But it sort of is restricted to this expanded wave of humans that left Africa around that time. And we know that to be true. It's because every non-African population, whether they're from Europe or from East Asia or from Oceania, so PNG or Papua New Guineans, everybody has a certain percent of Neanderthal ancestry, and that's been quantified to be about 2 to 4%, on average about 2% across all non-African human populations. I will say that for some of you who may be a bit more into the literature, certain African populations have also more recently been shown to have Neanderthal ancestry, but most of them are from the northern and eastern parts of Africa, and that's because of more recent sort of gene flow from Eurasia back into Africa, which has introduced the Neanderthal genes into these populations. So I just wanted to kind of PS there for those of you following more closely. So this was sort of a massive story to come out, because prior to this paper in 2010, the sort of prevailing thought from earlier genetic studies that were done not on genome-wide data, but from mitochondrial DNA data, was that in fact there had been no admixture between Neanderthals and humans. So this really has radically changed the way we think about our species and how our migrations also brought us in contact not just with other human populations, but also with other forms of humans and hominins. And the same year, in fact, the same group, collaborating with a group in Boston, found evidence for a second, admixture event with another form of humans called the Denisovans. And what's really unique and cool about the Denisovans is that this is probably the only form of hominins we know so far that hasn't been described from fossil evidence, because we don't really have a full-blown sort of skeletal evidence of Denisovans anywhere. And they've been essentially described for the first time based on ancient DNA or genetic data, and that came from this little piece of bone from that's almost from a pinky finger here. And it comes from this cave called the Denisova Cave in Siberia. So the name of the sort of hominin form was named after the cave in which the remains were formed first. Again, there was DNA preservation. And what the research team found was the Denisovan genome sort of clustered closely with Neanderthals. So there seemed to be related to Neanderthals much more than either of them are to modern human populations. But more interesting, when they kind of did a similar analysis to look at whether human populations today have ancestry from Denisovans, they did find that just like how all non-African populations today have ancestry from Neanderthals, certain populations of the world, particularly in Oceania and Melanesia, have ancestry from Denisovans. A few years later, certain teams also found that a small proportion of Denisovan ancestry is also present in many parts of East and South Asia. But the strongest signal that we see, up to almost 5%, is in fact found in Oceania. And this sort of really brings to light that as humans are moving around, they're encountering whoever was present at the time. And these migrations have really, brought us in contact with and shaped the way that our genomes are kind of, retaining legacy of these extinct hominin forms in many ways. And of course, also how these are sort of patterned in some ways, because, neither African nor European populations have any semblance of Denisovan introgression. This is restricted mostly to southeastern and Oceanian populations. On the other hand, again, Neanderthal legacy has been primarily impacting non-African populations. So again, it sort of goes to show how important migrations are in sort of driving genetic variation in different human populations. Okay, so now I want to come to our species. So while here sort of we're looking at sort of the more ancient picture, what happens when we met other hominins, but what happens when modern humans sort of meet each other, so let's kind of get into more recent histories. And again, I'm going to take a few snippets from different regions of the world because we have limited time, and I want to make sure I cover a few stories here that we can then open up for discussion. So I picked a couple of case studies from Africa to begin with. As I said, Africa definitely has the most deep population structure in all of human populations. And in fact, some of the Southern African traditional hunting and gathering populations, like the Khoisan, are regarded as some of the deepest genetic lineages in all of human population histories. And you can see some of these dates to almost definitely well over 200,000 years. There's a little bit of discussion around the timelines based on genetic analyses, but it's what's not under debate is the fact that some of the deepest lineages are found in Africa today. And we, again, because of the lack of a lot of ancient data from 50,000 years or even deeper than that, given how deep this the history in Africa is, the lack of ancient DNA data really limits us from making fine scale or fine resolved inferences about what human population structure and genetic variation look like deep in the past. So a lot of our sort of understanding for a long time about population histories in Africa came from modern genomes alone, but that changed particularly for more recent times in the last sort of 10,000 to 15,000 years. Now we have in the past about 5 years or so, in fact, overturned a lot of the prevailing thought in the field that we would never get DNA from Africa because it's so hot. But in fact, a lot of teams have leveraged technological advancements, both in the way that we can extract and sequence DNA, as well as analyze DNA that's been heavily degraded. And so this has really shed light on more recent histories across Africa today. And here is a summary figure from a really nice review that came out now in 2020. So the field has moved on since then, but I thought that this is a really great summary figure to really kind of underscore how population structure and migrations have kind of shaped population histories in Africa. So what the authors are showing here is here's a map of some of the genomic data that they're considering in their interpretation and analysis. The circular symbols are all modern population data sets. The diamonds are all ancient DNA data sets. And the different colors represent the different regions of Africa. So blues are Northern Africa as well as the Middle East, so a little bit into Eurasia. The grays are sort of the western side. The dark green is Central Africa. The sort of bright green is Eastern Africa, and the reds are Southern Africa. And on the bottom here are shown principal components analysis plots. So these are essentially two-dimensional plots, which can really summarize the complex data that we have in the form of genetic data. And sort of the simplest way to think about this is, two symbols that are close in space on PCA likely share some sort of genetic affinities. Obviously, PCA is very, it's more high level, so it's helpful to think about pattern observations from PCA, which can then be tested using more statistical and other population genetic methods. But for the sort of sake of this, the message that I want to give out here, some of the salient features of, if we just focus on this PCA right here, is that there's a lot of genetic continuity over the last 8 to 10,000 years, which is sort of the time period coverage of the ancient DNA data here in this analysis, because as you can see, a lot of the diamonds, which are the ancient DNA, fall on the same color circles. So that essentially means that a lot of the structure in present-day populations in Africa sort of were likely formed a few thousand years ago already. Of course, this is a broad scale sort of takeaway, and there are still certain events in more recent history, such as migrations, that have shaken things up. And just to kind of give you a sense of that, these studies have particularly found that despite the sort of more broad-scale genetic continuity, there have been migrations both into Africa over time as well as within Africa. For example, some of these migrations into, a few thousand years ago into East Africa have been proposed to, in fact, be associated with the expansion of potentially agriculture or pastoralized subsistence. So these probably brought in either agriculture or dairying into the region. And likewise, there's been expansions, within Africa as well. And one example, which is this green arrow that I wanted to talk a little bit about, is the so-called Bantu language expansion. And so the Bantu language family is one of the largest language families globally today. So you can see the range. It's sort of spoken all over from west to east to southern Africa today. And there have been, this is a very, very well characterized migration that happens about 4,000 to 5,000 years from this region of West Africa and sort of spreads rapidly, at least the language spreads rapidly across this broad swath of Africa. And one of the questions has been whether, whether the language spread was mediated by people moving, or was it simply sort of, the technology or just the language moving without the movement of people. And this has been answered again, time and again, both using ancient DNA as well as modern DNA. And in this particular summary, I thought they show really well that when you look at, so the reds, remember, are South Africa, or Southern Africa. So you have, on this PCA, you have the clustering of modern South African populations in the red circles, along with more ancient samples about 2,000 years ago, and all of them sort of clustered together here. But a few individuals who, if you can see right here, they're kind of hidden behind this red diamond. So these are gray diamonds. They were found or they were excreted in Southern Africa. But when you look at where they cluster, they in fact cluster more with the gray circles, which are all kind of West African populations. And this essentially has been taken as evidence with many other genetic analyses that many papers have done over time, to suggest that populations or peoples from West Africa, in fact, moved and mixed and left their sort of genetic legacy in these regions, and this is likely, mediated the spread of languages. So the spread of the Bantu languages has been essentially accompanied by human movements and humans taking the language with them as they spread across these regions in Africa. Similarly, we've got now we move to Europe, and we've I believe you've had some exposure to perhaps the European population history with Dr. Regis Narsimon's talk, but I just wanted to kind of find parallels here, since we just talked about these Bantu-related migrations in Africa. In fact, even in Europe, over the last 5,000 years, there have been this one key migration that has been characterized both archaeologically and now genetically as well, where sort of the farming communities of ancient Europe, essentially mix with migrating pastoralist populations from the steppe, so further east, who kind of migrate into Europe about 5,000 years. They mix with the farmers, and sort of the Bronze Age of Europe starts with this mixed sort of gene pool, and this sort of legacy you can find of the so-called steppe herders. So that's in green here, that the culture is archaeologically called Yamnaya, and you can see across modern populations, many modern populations have this steppe ancestry at varying levels. Definitely you go, in fact, it's a gradient from North Europe where this ancestry type is maximized to more Southern Europe where you don't get a lot of in fact, the admixture isn't as pervasive as it is in North. But what's kind of interesting in terms of if you want to find parallels between the Africa story, the Bantu story in here, is that in fact the Yamnaya migration, the steppe migration into Europe, has been proposed to be associated with the spread of Indo-European languages into Europe on the one end. And in fact, more recently, Dr. Narsimon's paper has shown that there may be some associations of these, not this particular steppe group, the Yamnaya, but other steppe groups that went into perhaps West and South Asia and took some of these Indo-European languages into that region. And so, again, in terms of finding parallels of human migrations and how they've impacted our cultural histories as well, again, it's very important at this point to acknowledge that genetics is in no ways a direct, informant of what languages people speak, which is why it's very important to refer to these as associations. So the genetic ancestries that we see of the steppe people in Europe does in large part overlap with populations who speak Indo-European languages, and hence that association is taken at this point to imply that perhaps these steppe groups were the ones that brought Indo-European languages in. And in fact, the other technology, if you can call it that were brought by the Yamnaya because they were pastoralists in the steppe, is the process of dairying or milk drinking into Europe, and we'll actually revisit that in a bit towards the end. So to keep in mind about this association with dairying and Yamnaya into Europe. I think this is it's a little old, this figure, but I think this is a really nice figure that summarizes, particularly because we have so much data from ancient Europe and modern Europe, and really summarizes kind of the impact that migrations and mixing of genes across different cultural phases and groups of people in the past have had. So essentially, you can this is a metric. It's called an FST for the population genetics and geneticists in the audience, but for the rest of you, just think of this as indicators of genetic differentiation between different groups of people or different individuals. And so we're going from, so these are 4 rows of data, and each row is a different time period. We go from the from pre-Neolithic, so sort of the before farming in Europe, to the Neolithic and Copper Age, so when once farming and domestication are introduced, to the Bronze Age about 5,000 years ago, and to modern populations. And one very striking kind of pattern you can see here is that the level of genetic diversity or differentiation among people in each of these sort of time periods essentially goes down, right? So you're losing the differentiation factor, and one of the key drivers of this sort of almost homogenization process has been migrations and mixing that have really brought differentiated groups, genetically differentiated groups into contact and be able to kind of exchange genes with each other. Great. Okay, so let's jump again. I'm sorry to make you travel so much across the world, but now I want to kind of spend a few minutes on the Americas. I know you had an amazing discussion with Dr. Maria Avila, who went through the paleogenomic research that her team and other teams have done in Mexico. So I want to kind of zoom out a little bit, because this was also work that I did as part of my PhD in Denmark. And this sort of concerns the peopling, the earliest peopling of the Americas, and thinking about sort of the founding populations of all indigenous communities across the Americas. And when we started this work, there was already a consensus in the field of genetics and in archaeology that Americas were peopled from Asia. And many of the other sort of proposals of peopling of the America have not found as much support anymore. However, when we started to sort of, or when my sort of involvement in this project came about, there was still a lack of information about who were the ancestors back in Asia who sort of, eventually gave rise to the founding populations of the Americas. And in 2012, there was this very interesting paper, and particularly this figure that was very relevant to us a few years later when we were trying to tackle this question. So this is a plot which essentially it's, one can think of an sort of almost like an admixture plot. So it's sort of kind of, considering different contributions of different lineages in the past to modern populations in this case. So in particular, I want to draw your attention to here, Europe and Americas. And in this particular case, this group, in fact, they published a suite of methods in this paper. So it was a methods paper where they published a now very commonly used set of population genetics tools that ancient DNA people in particular use to model admixture between within different ancient groups and modern groups. And so in publishing this method, they proposed this sort of scene where they said that there was some ancient North Eurasian, and that you can notice here's a question mark, because they didn't have any data from this. So this was basically what we today call a ghost population or a statistical construct. So they needed to statistically introduce some kind of a population in the past, which contributed on one hand to indigenous American populations and at the same time to Northern European populations. And at that time when they published, again, like I said, there wasn't any empirical data on whether such a entity existed in the past. And luckily for us, when I started my PhD, we didn't know at that time, but eventually as we did the analysis, it became clear that we had found this ghost lineage of what contributed to the founding populations of the Americas. And this was from a site called Malta in southern Siberia, where a 3 to 4 year old child was excavated. And this was, it was dated to about 24,000 years ago. And very quickly, what we found was the genetic affinity. So this is, again, a PC of this individual 24,000 years ago, are sort of between Europeans and Native American populations. So very similar to kind of that graph. And I'll skip quickly to the model here. And when we model the genetic ancestry of this Malta individual, we found that the lineage of Malta, in fact, contributes about almost a third of ancestry to Native American populations. In this case, we took Caritiana, which is an indigenous Brazilian group. But in fact, we found the Malta affinity to be across very similar and across many, many indigenous communities, all the way from Greenland to South America. And so this led us to kind of propose this model, which is very similar to the ghost model that David Reich's group had proposed a few years back. But in this case, now we actually found the identity, in fact, of this ancient North Eurasian, and that was through this individual that we sequenced from Malta, the site. And over time, there have been, I don't have time, but over time, many colleagues and other people in the field have really dissected migrations within the Americas. And I'm sad that I'm not going to have time to discuss that, but I'm happy to share references if anyone is interested. I have maybe 5 more minutes before I hand it off to Mashaal. So I just want to quickly take you to the last region that I want to discuss, which is South Asia. This is the region where right now my group is doing a lot of work, both from ancient and modern population data. So here, this example, which actually just came out yesterday, it was published yesterday. And here we essentially only at this point integrate modern DNA data. And here we study, in fact, the role of early migrations into South Asia, because South Asia sort of is on one of the proposed routes out of Africa that goes into Southeast Asia and into Oceania. The routes are still very much contested. Not a lot of evidence has been found, which is why it's kind of interesting to think about some of these coastal regions when we think about perhaps where some of the earliest members of the out of Africa wave may have potentially gone through. In particular, the role of Sri Lanka has been, which is the island nation of India, has been very, very interesting, because today they have a group of indigenous peoples called the Adivasi, who are proposed anthropologically to be descendants of the earliest some of the earliest sort of humans who were in South Asia many years, thousands of years ago. And contrast that to Sinhalese, who are today the most urban. About 75% of Sri Lankans self-identify as Sinhalese. So these are ethnic communities, and their oral histories, in fact, say that they did migrate from India more recently within the last 2,000 years. So there are these two very contrasting groups. Let me skip ahead to the admixture models that we kind of found, in fact, say that the Adivasi, so this is sort of, these are called admixture plots, where every color represents a different source. So we have 3 sources. The blue one is an ancient hunter-gatherer. The red one is a Neolithic. It's related to Iran, and the yellow one is related to the steppe. So very similar to the European steppe. As I mentioned, there's also been steppe migrations into South Asia. And you can see that many Indian populations that we took as example, as well as the Sinhalese and Adivasi from Sri Lanka, in fact, have a certain proportion of all these components. But more interestingly, what we found is the Adivasi have a higher proportion of the hunter-gatherer blue ancestry. And this, along with many other analyses that I wouldn't have time to explain too much today, has led us in this paper to propose that the Adivasi were likely the descendants of the early hunter-gatherers, but then later these groups like the Sinhalese, who came from India with all these other, the red and the yellow, sort of then mixed and diluted the blue ancestry in the Adivasi. And this is something that we really are interested now in investigating from ancient DNA perspective, and are working with collaborators in South Asia. So I see that we are almost at time, so I just want to skip on to my summary slides. Let me just do that. Oops. So In summary, basically, I want to impress upon the fact that migrations and genetic admixture have been very, very common in our species history. We can use both modern and ancient DNA to uncover migrations that have happened at different spatial and temporal scales, as we saw some examples today. And they've really, migrations have, as we saw today, again, impacted regional population genetic histories and structure, as well as potentially cultural histories that again, we've seen sort of perhaps associations with the spread of a certain subsistence or language, family or language type that is spread. With that, I want to close and thank you all. And I will, as I stop sharing, want to make a key point, which I hope we can discuss, which is that human migration histories are not in isolation. When we think about them, when we work as scientists, we need to be ethical in considering the societal and broader impacts of these stories and of these events that we're trying to reconstruct, and work closely with communities as well as other fields to really make sure that the genetic evidence is giving a more true representation of the ancestries and migration histories of many, many communities across the world. So with that, thank you very much. I hope to open this up to Q&A or just discussions, and would love to hear any comments. Over to you, Mashaal.
Great. Thank you so much, Maanasa. Muchas gracias. That was a really interesting talk, and is really bringing now to case studies. And so it's completely lining up with the cycle as we've seen it so far. So I saw that I'll start by asking about lactose resistance. I saw that you had something that you skipped. Yes. We heard about it from Vajish. And so I'd love to hear what you were finding about it in South Asia, to give the listeners a perspective on that as well, since they mostly hear about it with respect to Europe and so on.
Yeah, absolutely. So, the history of lactase persistence in South Asia is very interesting, because what I didn't have time to get into is South Asia has a high what we call very strong population structure. So we have ancestries that are represented both from sort of Eastern Eurasia as well as Western Eurasia, as well as indigenous ancestries from tens and thousands of years ago. And so the distribution of the lactase persistence as trait, but also in terms of the genetic variation, is so far the surveys have shown that it's closely linked, and in fact the variation, the lactase persistent variant that we find in South Asian populations is the same as the European one. Right now, the proposal is that the spread of this variant into South Asia is linked to the spread of the steppe ancestry, again related to Indo-European languages. So it's very similar and paralleling to Europe in many ways, and that is currently absent or in very, very low frequency in populations, for example, from the eastern part of the southern part, who retain ancestries that are more indigenous compared to the steppe-related signal that came in, which is more pervasive in Pakistan, northern India, and so on.
Okay, great. Thank you. So we have a question. So we have a lot of questions. Let's see what we can get through. So one of the questions is on what are the ethical considerations in using genetic data that has already been collected but under suboptimal ethical considerations?
That's a very good question. I think there's some discussion about this in the field, but there isn't really much regulation, because when someone studies and publishes, the data is out there. It's sometimes very hard. In fact, some of the legacy data sets that we've had from some of these big consortium that are pervasive now in many, many human genetic studies may have had fairly, sorted histories in terms of how they were collected. In fact, even when you think about ancient, human remains, a lot of collections may come from colonial collections. And so it's, on the one hand, these do form particularly since the question was about data, so I'll address that. The data is very important, because without contextual data, it's very hard to interpret any new data that we may be adding on. On the other hand, yes, absolutely, I think we need to be cognizant of the fact that our field has been built upon a lot of, unethical ways of collecting and doing science, and this has gone all the way back to the 50s or even earlier. And so, a few ways, there could be many ways one can think about in terms of how to address this. One is to think about potentially sampling anew with new engagement, with more proper engagement with some of these communities. As long as, again, they haven't been, completely, they haven't grown to mistrust genetics because of these historic unethical events. The other possibility is to, if we have information, go back to the donors, although that may always be somewhat challenging because many donors, particularly if these are legacy data sets, may either not be, even alive to speak to their own interests, or it may be very hard to identify and go and make a case. So I think definitely these are among many ethical considerations and issues in the field, broadly in population genetics, but particularly in ancient DNA, that we need to discuss and resolve. So thank you for that question.
Well, thank you for the thoughtful comments. I think the idea here is, like with anything else, like we have to keep moving forward. We have to reflect on how things have been done not well, and we have to sort of just move forward trying to do them better, right? So I think that's in general what human genetics and science overall is trying to do, and I think is being reflected in this cycle as well. So we have some questions related to population replacement and so on. So I'll just put a few of them together. What factors drive population replacement versus only cultural assimilation? And a related question is, agriculture generates population expansions. Do we always find population replacements when they encounter hunter-gathering populations?
So this is a complex question because it's hard to always know, particularly when we're looking at genetics. In fact, purely coming from a genetics background, I'd be very hard pressed to know the motivation of groups, particularly in the past, who moved. Of course, one has to integrate other fields to fully start to understand what may drive someone to move from one place to another, instead of simply sort of giving their technology away. And again, these are very much speculative at this point. Obviously, like I mentioned in my first slide, people may simply move because there are better resources. Maybe they've exhausted resources where they are, or they want to explore and find out what's better somewhere else, or purely from an expansion perspective. Particularly when it comes to agriculture, one thing is clear that again, genetic data and archaeological data both support that agriculture has been one of the strongest drivers of human population sizes. So human population sizes expanded massively during the Neolithic. And so what were originally small groups of hunter-gatherers and foraging societies who could essentially, sustain in certain regions, maybe, partake in the resources and move on to find more resources. The expansion of agriculture, in fact, required more space to accommodate the density of populations as human populations were increasing exponentially in population size. So they needed more space, needed more resources to do the agriculture. And so a part of that, large part of the expansion may have been simply to accommodate these growing population sizes and requirements of more resources. Hopefully that answered both questions in one. Otherwise, I'm happy to clarify.
Great for those thoughts. Thanks for those thoughts, Maanasa. So we also have some questions related to Neanderthal integration. So is it true that human to Neanderthal integration resulted in Y and mitochondrial DNA in Neanderthals having those parts from humans? If you can comment on if there was integration in the other direction and if that has been studied.
Yeah, so very succinctly, yes, it has, and there are papers that have characterized the other way as well, it's a little challenging because sort of in this case we have bidirectional, so definitely there's been Neanderthal into humans, and the at least a couple of papers have found the other way as well. But it's always a challenge because Neanderthals and humans also share DNA in more recent time. I mean, our separation has been in the last 500,000 years. So to be able to use a very, well-resolved ways to identify fragments or regions of the genome that have gone from one to the other and the other to back does require, and I think the reason why we've been able to identify this bidirectional gene flow is because now we have access to so many Neanderthal genomes, so we can directly compare differentiated parts of genome of Neanderthal with humans and be able to characterize whether there's been which way the introgression has gone. And yes, the evidence now suggests it's gone both ways.
Great. We also have a question on if there's any evidence of hybrid incompatibility between different human populations, the way that we see it for Neanderthal human hybridization events.
Oh, that's an interesting question. Nothing comes to mind because particularly when we think about hybrid incompatibilities, we're thinking about quite differentiated, more sort of on a species scale. When we think about modern human populations, differentiation, I think I may have seen, maybe John November alluded to that a little bit, that the differentiation within human populations is very, very minuscule in comparison. Our, what we call FSD or differentiation values are extremely low, which essentially is a product of our differentiation history or our species history only being in the last 50,000 years. So hybrid incompatibilities don't really play into these timescales and levels of differentiation among human populations.
Great, thank you, Maanasa. So basically, I think you're showing what we have been seeing throughout the talks in the cycle, that really amongst humans, the kind of diversity that we see does not, people have looked for these kinds of things in the last 100 years throughout, and they just have not found anything. In fact, the most recent studies are showing that in mixed individuals, such as those that we may found in Mexico that have recently got an introduced ancestry from different sources in Europe and Africa, from indigenous groups, we find that the genetics the variants that they have that may predispose to different diseases have effects that are very similar across those different ancestral backgrounds. They're highly correlated. So over and over again, genetic studies keep showing that it's really interesting to think about and study our diversity, and to understand our evolutionary history, but that we're not seeing that biological basis of traits or diseases highly different. It's more the interesting parts of adaptations to specific local contexts and so on than sort of grand differences between groups. So that leads to another question on, beyond skin color, what other seemingly superficial human traits have a genetic basis deeply linked to adaptation to different environments throughout evolution?
I mean, quite a few. In fact, I had a slide on that, but I had to skip ahead. But we talked about lactase persistence. We can also think about, in fact, a very interesting recent example has come from the Tibetan plateau, and that's actually through introgression from Denisovans into Tibetans, and that's a very local form of adaptation to the high altitude and hypoxic environment in Tibet. But we have several cases where in fact, for example, the Inuit of Greenland adapting to high fat diets, so that's sort of more a cultural induced adaptation to the sickle cell and malaria resistance in Mediterranean and certain African populations. And in South America, we have the arsenic exposure related adaptation. So there's a myriad. I mean, we could go on and on about how, and in fact, that's a natural kind of hopefully end to this talk on migrations, is that migrations really exposed our species to very, very diverse local environments, and that have really challenged our biology to adapt to these local environments. And so a lot of, many of these interesting differences we see among populations are in fact induced by natural selection and the need for these populations to adapt to specific environments. So yeah, skin color, hair color. So we have all sorts of pigmentation differences, again, depending on the latitude, and we have the vitamin D and several other hypotheses. But beyond that, some of the things I mentioned related to diet, related to pathogenic exposure, and related to environmental toxin exposures, or even the environment itself, like hypoxia and high altitude. So several examples.
Great, thank you. And similarly, if you can just comment on this, we have a question on, if we consider that most mutations are neutral or deleterious, how could natural selection operate so efficiently to give rise to the complexity of human characteristics that we observe today?
So one, there's still a lot of work being done in this field, and in terms of trying to understand. But there have been some succinct examples, particularly interesting ones that have come to light through ancient DNA as well, which suggest that many of these variants may have been neutral for a long time in our species history. Right. And at some trigger point, whether that trigger was caused by, a certain cultural or lifestyle shifts or what have you, essentially, brought or contextualized that neutral variant into a more challenging context, which then may have, shifted their essentially their value from neutral to something that helps adapt into a selected or positively selected zone in some sense. So this is sort of what's called selection on standing variation. It's not a de novo or a mutation that just appeared out of nowhere. So that's one way to think about it. Another way to think about it is also what we discussed in the context of these steppe migrations. I mean, these mutations may have existed in other adapted populations and then were brought into new regions through gene flow and admixture. And again, when they brought into new environments, how they sort of how they fared would have been again a product of the lifestyles and the cultural histories of some of these populations. In the case of Europe, these pastoralist communities came almost like the genes in this case also with the genetic variant for lactase persistence, as well as the subsistence related to dairying. So it sort of kind of came as a package in certain ways. So there are different ways to think about how a variant could enter either anew or be already present, but in a different context, and then sort of kick into selection mode through some drastic change in the environment or lifestyle of the community or the population.
Great, thanks, Maanasa. And what your answer also just generally points to the fact that variants never act on their own. Genetics is always acting within the context of environments which are always changing, working also in the context of genetic backgrounds which are always changing as migrations happen, as people mix. So in that sense, we cannot think of these things so simply, but it is a complex process that is going on. So I think with that, we can close the sessions in the interest of time. Thank you so much, Maanasa. So it's been really interesting to hear of these case studies from around the world. And next week, we will be hearing from Dr. Priya Moorjani from the University of Berkeley. La siguiente semana vamos a tener la doctora Priya Moorjani de la Universidad de Berkeley, hablando más sobre estudios locales en diferentes partes del mundo. Muchas gracias.
Muchas gracias, Jaime, muchas gracias, Mashaal, y muchas gracias, Maanasa, por tu presentación tan fantástica. Muchas gracias a todos. Resumo los pronunciamientos del doctor Jaime Urrutia. Y yo creo que lo que acredita el estudio Maanasa es que la migración pues casi es consustancial al hombre. Y las barreras que se construyen para esta movilidad pues suelen siempre ser barreras artificiales. Al final el hombre avanza, el hombre se interrelaciona, el hombre genera nuevas oportunidades. Y yo creo que lo que ha mencionado Jaime, pues es un sentir que recogemos todos los universitarios y que subyace en las preocupaciones de lo que ahorita está ocurriendo en algunos países, en Estados Unidos señaladamente. Algunas autoridades están bloqueando la migración. Esta presentación de hoy ilustra claramente lo que decíamos. Los estudios alrededor de la migración son cada vez más importantes y habrán de ser cada vez más dinámicos. Que la población es más numerosa, se mueve con más facilidad, se mueve con más agilidad. Seguir los rastros de esas civilizaciones, eso es algo que se plantea como un desafío científico muy relevante y para lo cual pues estamos convocando a todos nuestros jóvenes a que sigan participando activamente en los estudios relacionados a esta parte evolutiva, a esta parte de la genómica y de la paleogenómica y a esta parte que le sigue el trazo a los movimientos humanos que se desplazan no digamos de un continente a otro, sino de un lugar a otro en todo el planeta. Muchas gracias a todos, muy buenas tardes y seguimos contando con ustedes en esta serie tan relevante que estamos escuchando y que ofrece cada vez horizontes más importantes, más interesantes para conocer mejor cuál es el origen, el destino y la evolución de la raza humana del futuro. Gracias a todos, gracias Maanasa, gracias Mashaal, gracias Araxi, gracias a todos, muy buenas tardes.
Bienvenidos al ciclo de conferencias de investigación Universidades por la Ciencia, organizado por Fundación UNAM, el Colegio Nacional y el Consorcio de Universidades por la Ciencia. Les agradecemos el interés y que nos acompañen. Y el día de hoy tenemos una conferencia a cargo de la doctora Maanasa Raghavan.
Hemos becado a más de 1 millón de estudiantes universitarios.
Más de 84,000 alumnos y alumnas han sido beneficiados con becas nutricionales.
Lanzamos una campaña para apoyar a la Universidad Autónoma de Guerrero, afectada por el huracán Otis.
Este año organizamos nuestra tercera gala musical y nuestra cuarta carrera atlética.
Estamos felices porque más del 87% de nuestros becarios son la primera generación de universitarios en su familia.
Cada año se apoyan a 100 estudiantes de la Facultad de Derecho.
Hemos entregado más de 600 becas a estudiantes de bachillerato junto con Fundación BBVA.
Hemos transmitido 195 documentales y 160 conferencias.
Apoyamos a la Unidad de Investigación en Neurodesarrollo de la UNAM.
Todos nuestros informes son públicos.
Se han entregado más de 50 premios para fomentar la investigación científica.
Con el programa Bécalos, el Patronato de la Facultad de Química y Fundación UNAM otorgaron más de 30 becas.
Con la ayuda de Fundación Carlos Slim hemos otorgado becas de apoyo nutricional y económicas.
Con el programa Hídrico Modelo ayudamos a 340,000 habitantes.
Tenemos 2 premios EFI Social por nuestras campañas publicitarias.
Junto a Grupo VAL, más de 2,000 universitarias han sido beneficiarias.
Otorgamos más de 400,000 tratamientos.
Durante la pandemia apoyamos con las campañas Dona un Kit y Apoya a un Residente a estudiantes de medicina de la UNAM.
Durante el confinamiento COVID, donamos tablets para estudiantes de escasos recursos.
Hoy son más de 27,000 solidarios impulsando la educación.
Participamos en la conservación de corales en el Caribe mexicano.
Brindamos más de 4,000 becas de movilidad internacional.
Colaboramos con el Semillatón a preservar el maíz en la Sierra Tarahumara.
Apoyamos el proyecto de conservación de perritos llaneros en la Reserva de la Biosfera de Jano, Chihuahua. Participamos en el proyecto de conservación de la guacamaya roja en el Parque Nacional de Palenque.
Desarrollamos el programa Líderes Transformadores.
Participamos con el Consorcio de Universidades por la Ciencia.
Organizamos 11 ediciones del Foro 2020.
Fundación UNAM resguarda el Palacio de la Autonomía, inmueble histórico de la universidad.
A través del CI ofrecemos cursos, diplomados y talleres. Dotamos de equipo al Observatorio Astronómico Nacional de San Pedro Martes.
Apoyamos a alumnos de escasos recursos.